Welcome back, everyone. Today we're going to be learning about the structure and composition of the hair. So if you could take this picture here that I've drawn, I can tell you I'm no Leonardo da Vinci when it comes to drawing biology. But what we have here is, if you could imagine a strand of your hair just cut here in half, we would see that we have three different layers to the hair. We have the innermost layer, which is the medulla. Then we have the cortex, and the cortex here actually makes up a large part of the hair. And then we have the cuticles that are all along the outside, and we'll be talking a little bit about each of these layers. So starting with the, let's work on the inside out. So starting with the medulla. So it's in the middle of the hair shaft. This innermost layer is composed of round cells. There are some hair types that don't have a medulla, such as natural blonde hair. For our purposes as hairstylists, we are not overly concerned about the medulla, as salon services really don't involve anything to do with the medulla. So we don't have to be so concerned about it. Looking at the cortex, this actually makes up 90% of the hair's weight. And as hairstylists, what we do in terms of chemical thermal processes involve a lot of stuff with the cortex. If we were to put a perm or a relaxer into the hair, you are directly getting that cortex involved. And that's something that we're going to be discussing in further videos about when we do those processes, how that affects everything in the cortex, etc. Let's talk about the most outer layer, which is the cuticle. Now, the cuticle of the hair is like a scale or a shingle, and what they do is they just overlap each other. They are transparent cells that can open or close. The cuticle opens and closes when we form stuff like a permanent hair color, and it's really all about the pH levels that we're using at certain times to make the magic of hair color really work. For example, an alkaline such as hydrogen peroxide mixed with a hair lightener, this helps make the hair swell, and then the cuticles will be able to open up, and color molecules will then start getting inside these here, and as the color molecules go in, they deposit right into the cortex. So let's talk about the elements in the hair. So as you see, I have written here cones, C-O-H-N-S. All of these letters stand for an element. So C stands for carbon. O is oxygen. H is hydrogen. N is nitrogen. And S is sulfur. Now each of these add up to being 100% of the hair. Carbon is at 51%. Whereas oxygen is 21%. Hydrogen is at 6%, and nitrogen is only at 17%, and the smallest amount out of the elements is sulfur, coming in at 5%. Next, we're going to be talking about some of the smaller parts in the hair, and that is the amino acids. So amino acids, well, they join together and they form chain, and a bunch of them together of the amino acids, they form these polypeptides. And but they all bond together, these polypeptides. So there's very specific types of bonds for these. And the first one being the hydrogen bond, not the hydrogen bomb, the hydrogen bonds, salt bonds, and disulfide bonds. So we're going to be taking a look at each one of these in some further detail. So first, let's take a look at hydrogen bonds. So these bonds, well, they're broken down. They can be broken, they're a very weak bond. They can be broken down by water or heat. So it really doesn't take too much. When you use rollers, say in a wet set, you're actually stretching the hair, and when the hair dries, these bonds will reform, holding the shape of the roller. Again, these are very, very easy to separate. Now, the next ones are going to be salt bonds. These can be broken just by adjusting pH levels, and pH is something we're going to be getting into later on. Salt bonds break apart at a pH greater than 5.5. Now we're going to talk about the disulfide bond. This one's a little bit more involving than the other two bonds, and it is fairly different than the other two bonds. The salt bond and the hydrogen bond are both physical bonds. The disulfide bond, a chemical bond, and make no mistake, it is also a physical bond, but it is predominantly a chemical bond. So the disulfide bond, it forms between two cysteine amino acids. They're created by a process known as oxidative folding. Well, it's a complicated process about how they form, but what's important to know is that they don't break apart very easily. In a salon, we use a product that is called ammonium thioglycate, and it's used to break apart these bonds so that a new shape to the hair can be applied. And that's exactly how a perm works. Now, you can also use extreme heat to pull them apart. So, for example, if you're using a flat iron and you're pulling that through your hair, you're actually causing those bonds to come apart. But that is a very, very high heat that is doing that. But otherwise, under normal conditions, you got to use a chemical in order to get these bonds apart. Moving into the 3 growth phases. So there are 3 phases that your hair undergoes when it's growing, and the first one being the anagen phase, and that can last up to several years. And then there's the catagen phase, short duration, and then that will move right into the telogen phase, which is the final phase. So in the anagen phase, this is also known as the growth phase. The anagen phase, it dictates how long your hair is going to grow. So typically this phase can last anywhere from 3 to 5 years, but in some people it can last a lot longer, say up to 10 years. So if your hair grows half an inch a year, then at the end of 10 years, should you never get a haircut, you can expect to have about 6 feet of hair. Now, during this anagen phase, the hair is attached to the dermal papilla, which is the blood supply to the hair, is still attached to the hair. But then once this period is done, like I say, it could be anywhere from, say 2 to 5 years, up to like 10 years or so. But once that's all finished, then the blood supply starts to cut off. So the dermal papilla ends up disappearing. And that will bring us into our next phase, which is the catagen phase. So it's a transition period, and it only lasts a couple of weeks. And once the dermal papilla detaches, then you start to get into our very next phase, which is the telogen phase. So this usually lasts, once you're into that telogen phase, it lasts about 3 months. The hair has now lost its blood supply, so the hair can start to appear to be a little bit dull and lifeless. Now, fortunately, only about 10% of our hair at any given time is in this phase, and 90% of our hair is in the growth phase. So out with the old and in with the new. The old hair will stay in place in the scalp until one of two things happen. It'll either get jostled out through friction, such as, you know, when you're shampooing or brushing your hair, or it'll just get shoved out by the follicle producing a new hair in its place. But that is it for today, folks. Thanks so much for watching, and I hope you learned a little bit from this. And I look forward to seeing you guys next time.